Thanks for joining me in a session on autism, models, and treatments. I hope that you'll grasp by the end of this lecture how Timothy syndrome, a rare form of autism, serves as a model for understanding the pathophysiology of autism, and also recognize that CBD is a possible treatment for epilepsy and autism, and know a little bit about its mode of action. This is a young man, shown here at age 11, who showed signs of classic autism and epilepsy, with first hints at 3 months, and then language and eye contact regression at age 2. He had social aversion, inanimate object preference, and altered communication, and along with it, perseveration, avoidance of novelty, and stereotypical behavior. Harry was the son of my business manager at Stanford before I came to NYU. His case, like many other cases, is on the rise. The diagnostic cases of autism in the United States are now ranked at about 1 in 60, with an increase over the last several years. Genetics accounts for about 50% of the risk of autism. The rest are perhaps by environmental factors and factors we don't understand. Boys are 4 to 5 times more susceptible to being diagnosed with autism. And finally, there's a great variety in co-occurring conditions, including social interaction deficits, stereotypy, and particularly intellectual disability, sometimes sleep problems, and epilepsy in about a third of the cases. Timothy syndrome is a rare form of autism spectrum disorder. The children present with altered facial features and webbed fingers and toes, indicating abnormal development. They have an unusually long EKG-QT interval, immune deficiency, but most importantly, they have a 60 to 80% chance of developing autism. This has been traced to a mutation in a calcium channel, the same calcium channel that drives the heartbeat, the heart contraction. The mutation increases the amount of calcium entry into the cell, seen here as an increased area under the curve, and it also causes the channel to open at abnormally negative voltages, so that there's more opening of the channel. If you put this mutation in a mouse, you see some interesting resemblances to human behavior. On one hand, learning and memory seem to be normal. If you put the mouse into a water Y maze, it will start off behaving at chance, going 50-50 left and right. But over a few trials, it will learn gradually that turning right is the thing to do to get out of the swimming and onto a dry platform. If you come back and test a day later, both the Timothy syndrome animals and the wild-type animals remember to perfection. Now, if you test whether the animal can reverse its behavior, that's when you see the resemblance to autism. By putting the platform on the opposite side, we can show that the wild-type animals will gradually start off going what is now the wrong way, but eventually learning to land on the dry platform. That's shown in this movie here. The animal used to turn right, but now it has learned eventually to turn left, and it finds the hidden platform. Now, let's see what happens in the Timothy syndrome animal, where my colleague has put a plastic partition here in order to coax the animal to try to go to where the platform is. But the mouse stubbornly insists on turning right every time, continually traversing the maze and turning clockwise in a vain effort to get to the dry platform. And this was seen consistently in these animals, indicating that the single point mutation caused a mouse equivalent of the persistent behavior that we see in humans. Now, let's talk about CBD as a possible treatment. Work done here at NYU by Oren Davinsky and colleagues showed efficacy in treating Dravet syndrome, a syndrome characterized by repeated seizures and autism. As a result of this trial, a FDA approval was given for a plant-derived formulation of CBD. And in fact, clinical trials are ongoing at NYU to test this on the more general autism population to see if they can also be helped by this agent. Interestingly, if you make a mouse model of Dravet syndrome, there is an improvement in social behavior. The affected mice are much less sociable than wild-type mice, but if you use CBD, you can restore this back to wild-type levels. There's a lot of overlap between epilepsy, autism, intellectual disability, and other disorders such as schizophrenia seen at the genetic level. So there are also clinical trials going on in England using CBD with schizophrenic individuals. So how does CBD work? Well, I'd like to tell you a little bit about an endogenous signaling system that involves a lipid molecule called LPI. LPI is derived from a much more common lipid called phosphatidylinositol, or PI. That is a lipidic molecule with two fatty acid chains and an inositol head group. It is hydrolyzed to lysophosphatidyl'inositol by the enzyme phospholipase A2, a calcium-dependent enzyme. So as a result of activity, you split PI into LPI and a fatty acid chain. Now, interestingly, the back reaction converting LPI back to PI is mediated by another enzyme, LPI acyltransferase, putting an acyl group back on LPI. And that enzyme can be mutated, and when individuals suffer that mutation, they undergo epilepsy and autism. So this provides genetic evidence that the removal of LPI can be lesioned, and that is directly causative of the very disease that we're interested in here. How does the LPI act? LPI acts as an agonist for an orphan G protein coupled receptor called GPR55, and CBD, as it happens, blocks the action of LPI. So the idea is that LPI, if found in excess, can cause seizures, and CBD prevents this by preventing the action of LPI on this receptor. So the target of action is a 7-transmembrane G protein coupled receptor. So Evan Rosenberg, while he was working in my lab as an MSTP student here, came up with the following hypothesis. Normally, there are endogenous levels of the receptor GPR55. They interact with LPI, and together they tend to favor excitation, as he measured, and decrease inhibition. In other words, LPI and GPR55 step on the accelerator and release the brakes. This causes a tendency to overactivity, such as seizures. Now, Evan found that seizure activity actually influences GPR55 levels, increasing them, and further increase the level of LPI itself. This combination would make stronger signaling and further increase excitation and decrease inhibition. So this represents a positive feedback loop involving elevation of both the ligand and the receptor, and potentially this could contribute to autism and epilepsy. CBD blocks the effect of the LPI GPR55 and therefore prevents the overexcitation and inhibition. So the take-home points of this lecture are: Timothy syndrome is a rare but highly penetrant form of autism due to a mutation in the very calcium channel that drives the heartbeat. CBD has been used already to treat rare forms of epilepsy, is under clinical trials for general forms of epilepsy and also for a role in autism. Increased levels of LPI caused by a genetic lesion in the enzyme that removes it lead to seizures in autism, implicating LPI. And finally, CBD, by blocking LPI action on its endogenous receptor, may prevent the action of LPI and thereby combat seizures and potentially autism. Thanks very much for your attention.